Good afternoon to this, welcome to this afternoon's session. We have Louis Stagnetto, who's going to be given a presentation. Louis is the marine biologist and co-founder of the Nautilus project. He's a PhD student within the Institute of Life and Earth Sciences at the University of Gibraltar, with a particular interest in how climate change is affecting the oceans and the organisms within it. Louis.
Thank you very much. I want to pick up from where the last discussion left off about the vertical sampling throughout the water column. And this is something that, in conjunction with the university, as part of my PhD project, is something that we're actually doing. We're taking vertical transects of the water column down to about 80 meters. We are looking at temperature, salinity, oxygen saturations. We're looking at various variables in order to try and see what is happening in the waters, certainly in our southern coastline. And that kind of leads in quite nicely to what it is I'm doing, which is investigating the phytoplankton populations in the straits, trying to understand how they're changing and what's driving those changes. Are these changes due to physical changes in the water column? Are we looking at chemical changes, things like nutrients and nutrient limitation? Or is it biological? Is it predation? Is it viruses, etc.? So this is what we're trying to untangle. And this is to try and understand the productivity and the carbon sequestration that might be happening in the straits in order to be able to perhaps offset carbon in the future. So in order to achieve this, the talk outline, I'm going to give a bit of background to the research. For those of you that don't know about plankton, I'm going to give a plankton crash course so that we have an idea of what we're talking about and look at the significance, how they play out. We're going to look at some physics in the straits, really understand how dynamic this area is and how that makes it very difficult in some respects to sample. And then we're going to look at the chemical signatures that I've picked up and going to try and fit in some biological aspects to that. And at the end we'll have a question time. So this is Professor Paul Tyler from Southampton University. He's a very eminent deep-sea ecologist who did a lot of work with the hydrothermal vents in the mid-Atlantic ridge. This is his baby. He's a master at this. He took us for marine invertebrates, the first course in my first year at Southampton whilst I was doing my marine biology degree. And he sat there and he looked at all of us and he said, "So how many of you are here to study the dolphins?" And about a third of the room put their hand up, and then he went on to say, "Right, and how many of you are here to study the sharks?" And about a third of the room put their hand up. And then he went on to say, "How many of you are here to study the turtles?" And again, about a third of the room put their hand up. And he said, "For those of you that are seriously considering a career in marine biology, what you will be studying is this." And I remember sat there looking at the slide thinking, there is no way that that is more exciting than a shark. But as you begin to understand what is happening in the water column, and you start to understand what is driving, or try to understand, shall we say, what is happening within the water column and why these creatures are moving around, you start to get an appreciation for how important these marine plants are. And consequently, marine plankton suddenly becomes a very fascinating subject. So I hope to enthuse you with that today. So the plankton crash course, why does this matter? What are phytoplankton? These are autotrophic single-celled organisms, so they photosynthesize, they produce their own sugars. They inhabit the upper sunlit area of the oceans, which we call the euphotic zone. And they are an extremely diverse group of organisms. We have photosynthesizing bacteria, we have plant-like diatoms, we have heterotrophic dinoflagellates, and we have armor-plated coccolithophores. Although these species are somewhat related, there are more differences sometimes between the species than similarities. So actually, it's a very interesting group of organisms to study. The cyanobacteria are free-living. Again, they are photosynthetic. Their primary pigment is green, although they do have a green-red ratio. And they're considered to be the main organism responsible for the oxygenation of the Earth's atmosphere, certainly in early primordial history. They're dominant in both freshwater and marine environments, and they typically occur in chains. So when trying to identify it, that's really what you're looking for in the cyanobacteria. The diatoms are enclosed with what's called a frustule, and one part fitting over the other. So if you imagine like a little wooden box which has two parts, the way the two parts fit together, this is how this frustule fits together. And the frustule, rather than your wooden box, is made from silicon. So that's one of the signatures you can look for in the water column. They are dominant in temperate and cold regions, and they may occur in chains or as single organisms. Here we have some dinoflagellates. They are a major group of phytoplankton. They typically contain two flagella, and you can see the groove running around the middle, and this is where these flagella are normally found. This one is on an SEM scan, and so there's no living tissue here. But that's normally where the flagella would be found. It helps them, it makes them motile, it allows them to move through the water. They lack any external skeleton of silicon, but they are armored with plates of carbohydrate cellulose. And they are solitary single-celled organisms. And of that, Noctiluca and Dinoflagellates, they obtain organic carbon through ingesting, feeding on other phytoplankton species. So they're like the carnivores of the herbivores, if you think about it that way. Omnivores of sorts. Finally, we've got the Coccolithophores. These are my personal favorite. These are solitary single-celled organisms. They're distinguished by their calcium carbonate plates, which are called liths. And this picture here that you see, this is one that I took using a scanning electron microscope from the work that I was doing in Wilmington when I was doing a research project out there. And these cells, they're normally about 10 microns wide. So they're actually very, very small indeed. And they tend to bloom in nutrient-depleted waters, so typically offshore. And they're very important species in the study of climate change. And we'll see a little bit about this now. So over here we have the southern coast of the UK, Cornwall up to about Falmouth area. And this white milky area that you can see, this is actually the spring bloom of Coccolithophores as seen from space. So you can see that although this is a tiny organism, it actually has a fantastic effect on this localized area. And what they do is they produce these liths that we were seeing, the armor platings that you can see on the outside, and they continuously produce that lith, and the older ones start to fall off. And those liths start to sink in the water column. And so what I'd like to show you now is the video of how they actually produce these liths. And this is a video by Alison Taylor, who was my supervisor whilst I was in Wilmington. Cell. Okay, so these are calcium carbonate liths, which are now being exocytosed onto the outside. These cells were actually, they were decalcified by putting them in acid first, and this was as work where they were investigating the effects of acidifying oceans to see what would happen in climate situations. And so we can see how this cell is rebuilding the lith structure that surrounds it. Little is known as to why they do this. Some schools of thought say that this is due, they use it for protection. Others say they use it to control the amount of light. There are various schools of thought out there. So, what does the literature say about the area? So phytoplankton communities in the Mediterranean, what we see, Estrada et al. In their paper in 1999 studied the Alboran gyre, and what they found, as expected, was that in the Mediterranean we are dominated by diatoms along the coastal region. Diatoms like lots of nutrients, and so you tend to get the most amount of upwelling in those nutrients when you've got these mini gyres that occur as the currents move against the coastline. And that tends to bring nutrients up, which makes the diatoms very happy. They outcompete all other organisms along this area. And in the offshore area, it's dominated by Coccolithophores, which actually, as we said before, they tend to do well in the nutrient-depleted areas. On the other side, we have this is work by Estrada et al. That was done in 2016. This was using similar techniques to understand the Atlantic side. And what they found there was, once again, the diatoms are the dominant species, phytoplankton species along the coastline. We have the Coccolithophores as expected offshore, but we also have presence of Dinoflagellates. These were the predatory phytoplankton group, and flagellates too. So the complete picture looks something like this, with an obvious omission. And the omission is where I'm trying to understand what's happening, which is in the bit in the middle, because it's in the bit in the middle where we have the most dynamic action happening, we have the most mixing, and where we have the most questions as to what's happening in the strait. But first, let's look at phytoplankton and their significance with respect to climate change. And so this is from a paper from Nature Reviews, and I think this describes this very well. Anthropogenic CO2 in the atmosphere, what we need to understand is that it's always in sync with the amount of CO2 that you find in the ocean. So as we produce more CO2, we are increasing the level of CO2 in the ocean. And for a photosynthetic bacteria, this is kind of good news because you've got more sources of carbon to be able to photosynthesize. So typically, plankton do well under those conditions, other than if you exclude for a second the acidifying effect that carbon dioxide might have. These plankton go through their life cycles, and they eventually die, and that's where you get the particulate organic matter. So this is one pathway down. The Coccolithophores are producing their liths, and these liths start to sink. And when eventually they sink all the way down, this is known as the biological pump. And this biological pump removes these liths or any particulate matter that might be in surface waters, and it locks it into oceanic sediments long term. Another way that we can get this carbon locked into the oceanic sediments is through viruses, and the viruses actually break down the organic matter and dissolve it. And they are then consumed by bacteria in the microbial loop. And as the bacteria start to clump together, they die. That ends up like marine snow. It ends up getting locked up in the marine sediments too. And over a long period of time, although we're talking about tiny, tiny organisms, we get structures like this. Okay? So this is the White Cliffs of Dover, and this is primarily made up of Coccolithophore liths over millions of years through the primary production that takes place in surface waters. Closer to home, our Gibraltar is made in a similar way, using a similar process. Okay? There are other, I'm not a geologist, so it's not my speciality. There are other things that go on with limestone, but it's a very similar process that takes place. So, onto the work that we're doing. This is our sample site in the southern waters of Gibraltar, over 200 meters worth of depth at this point. Unfortunately, our depth finder on the boat doesn't go any further, so I can't be more specific on that. And what we do is we are sampling vertically the water column in order to try and work out any signatures, temperature, salinity, any chemical signatures, or even chlorophyll or pyrethrin signatures that might exist. And these last two are the photosynthetic pigments that exist. And so you can use that in order to find where your phytoplankton are. So let's look at the physics of the straits. Here we have a bird's eye view of what's going on in the straits. You can see Gibraltar in the top right, and what you see is a very deep channel. If you look at the key down the bottom, what we have is a deep channel of about 800 meters, which goes all the way past Tarifa, and somewhere in between Tangiers and Barbate, you get the Camarines sill. And this actually raises the bottom to about 300 meters, and it forms a bottleneck for water trying to either enter or leave the Mediterranean. And as a very rough schematic, what we have here is what the water is doing. We have Atlantic water, which is typically lower salinity and lower temperature, which is coming in through the surface. And we've got Mediterranean water, which is higher in salinity and higher in temperature. And that is being forced up by the Camarines sill, and then it can leave out through the Atlantic. And we have this effect called tidal pumping. So if you look at the bottom, what you'll see is we are going through a tidal cycle, going through from neap tides now up to spring tides. And what you see is this Mediterranean water pushing up almost into the surface. And then at the point of high tide, it pushes this wave, which runs right the way right the way past Gibraltar. And it travels. It's a wave with an amplitude of about 100 meters, an internal wave, and the speed of it is about 1.5 to 2 meters a second. It's most predominant at spring tides, but you can also see this effect in the neap tides as well. And if you look on the side, what we have there is the label AW. That's talking about the blue area at the top. That is primarily our Atlantic water that's making its way in. Just below that, you've got a green area, which is the modified Atlantic water. That's the one that's starting to mix. And just below that, you've got the Levantine intermediate water. And below that, in red, which is the highest salinity, we've got the Mediterranean water trying to leave the basin. So with reference to the waves that we were looking at, this is a picture from NASA in 1984. Here we see the straits from a bird's eye view, a photograph from a satellite. You can clearly see these tidal waves actually propagating out into the western Albarine gyre. Okay, so it's quite a beautiful thing to look at. You don't need to be in space. This is a photograph I took from our sampling site looking out to Jebel Musa. And if you look at the water in the foreground of the picture, it's nice and choppy. And if you just cast your eyes slightly behind that, you will see that the water's very flat. Go a little bit further, you see there's a layer of choppiness again, and just behind that, you see that it's nice and flat again. And so what you have there is the effect of the wave, the tidal wave coming through. Which means that when you come to sample, so this has such a great effect on sampling, that you really need to know what water you're sampling in before you can make any type of assertion as to what you're seeing. And so this is some data now from my PhD. What I've done here is I've used the definitions from Zavartelli and Mello from 1995. These are their definitions. I used those definitions as a guideline, and then I plotted my salinity plots against temperature to see where my water fit. And as you can see from the picture, the majority of the sampling for all those months from March 2019 all the way through to September 2020 sits quite squarely in the modified Atlantic water with a signature of Atlantic water in some of the samples. And this really helps to ascertain what sort of life you're expecting to see based on studies that we've already looked at. And looking back at the image we were looking at before, the video we were looking at, this is just an image, but what it shows you is that we are only in the top about 100 meters of water, which is the Atlantic water and the modified Atlantic water, which is just about right, because the probes, the length of cable that I have is 80 meters, and so it really does show, it ties everything up quite nicely on the physical. So what are the chemical signatures? So here we have photosynthesis. As many of us know, it requires light, and plankton must be in the euphotic layer. Okay, they need light in order to do it. Below the euphotic layer, there's not enough light. They cannot photosynthesize. We have more light in the summer, and typically we have less light in the winter. And certainly in the UK, this has a very dramatic effect. Over here in the Mediterranean, this is something that we need to see exactly what sort of effect it's having. But photosynthesis produces oxygen, and that is a trace that can be measured. And not just oxygen, but chlorophyll and pyrethrin, which are the two photosynthetic pigments, they can also be measured. And so this starts to lead in a little bit into some of the biology and what we might find. So here I have a plot, again, from March 2019 all the way through to October 2020. And this is looking at oxygen saturation. Now, you can't see it very clearly on here, but the blue line is the line that goes from, it's the bottom line if you're looking at the left of the plot, going up. And what we see are some peaks of supersaturation in the water. The y-axis is actually showing us oxygen saturation. The TV is going to automatically power off. Do I need to do anything? Do I press? TV is going to power off. Okay, just some technical difficulties. Brilliant. Okay. So 100% on the y-axis is showing us complete saturation of oxygen in the water column. And the reason why I chose this one to show you is because we have three events here where we have clear supersaturation taking place. Now, that effect, that degassing, could be a signature of photosynthesis. It could be due to warming surface waters, which changes the solubility of the gases. Or it could be a reduction in density or atmospheric pressure affecting the water column. So there are three things that it could be. But what I then did is I plotted turbidity next to it to see what effect turbidity would have. And so on the right side, you've got the axis for turbidity. The lower values are basically the water column is clearer, and as the value increases, the water column is filled with more stuff. And what we see is that within the water column, the peaks in oxygen saturation or the degassing is when the water column is generally clearer. In other words, there's the possibility for photosynthesis to actually be taking place. All right? So that's those 3 points over there. So now let's look at chlorophyll and how chlorophyll is affecting. So over here we have a typical plot for phytoplankton blooms in the Northeast Atlantic. And this is the sort of thing that I am trying to produce here for the Straits. So we understand what the species are, when they bloom, at to what level they bloom. So something like this. If we follow the all cells plot, we can see that there are two distinct blooms, one in spring and one in autumn. But if we look a little bit closer, we see that the spring bloom in composition is somewhat different to the autumnal bloom. So the spring bloom is dominated by diatoms. And then in the summertime, as the thermal layers start to stratify, we end up with the thermocline forming, and the plants are using up all the nutrients that are in the top layer. Lots of light, but that thermocline is forming a barrier which stops vertical mixing taking place. And so now we have nutrient-limited water in the area where there's the most amount of light. And so what we see is a shift towards the bacteria that are probably eating up the photosynthetic organisms that were present in those surface waters. And then in the autumn time, as the water starts to mix again, the thermocline starts to break down, we see a return of nutrients to those surface waters, and now we see a second peak in the northeast Atlantic. And this is typically made up of diatoms, cyanobacteria, which are the blue-green algae, and dinoflagellates, which are the predators. So here we have some work from Malaga University, and this shows what's happening between March and May. And if you focus in on what's happening on Gibraltar, you can see that in the area that I'm sampling in, we really are reaching the maximum amount of chlorophyll possible on their scale. And actually, the work that I'm doing suggests that the levels, the concentration of chlorophyll for our area is way, way beyond the scale that this shows. We can see the formation of the western Albarangaya, and we can also see the signature in the eastern Albarangaya there too. So I'll just draw your attention quickly. The chlorophyll concentration milligrams per meter cubed, and if we look across my plot, that's micrograms per liter. They are equivalent. Okay, so the top end of the scale for what they were measuring was about 1.5. You can see that at the top of my vertical transect, we have about 1. Okay, so it's within the ballpark of what they're showing. But they're only looking at surface chlorophyll. And as you can see, as you go down, so my y-axis is depth in meters, and as you go down through that water column, you see that in April, where we have a clear spring bloom, we've got concentrations of almost 12 micrograms per liter, which is an order of magnitude greater than anything that you're seeing from the surface plots. And looking in at that a little bit deeper, comparing chlorophyll and pyrethrin, the reason why I did this plot is because one would think logically that if it's one type of organism, the ratio of pigments within that organism would remain the same. So really what we're doing is we're looking for changes in the ratio to try and use this signature to tease out when we're having changes within the population. And if you look from January to March and in April, we actually see a very strong coupling between the two pigments. May, there is the end, what seems to be the end of the April bloom. And then suddenly in June, we get this signature over here, which is for, a dominance in pyrethrin taking place with what seems like, again, a slight bloom of something, the green pigment just below. And this picture was taken in that period. We were doing some surface samples. We found some pink scum floating on the surface. We took it back, we analyzed it, and what we found was that this was the dinoflagellate Noctiluca scintillans. It's commonly known as the sea sparkle. It's a heterotroph that engulfs by phagocytosis. It includes, its food is plankton, diatoms, other dinoflagellates. It even eats fish eggs and other bacteria. And what happens is that when it's feeding, you can often find diatoms actually inside it as it goes phagocytosing it. These were the images captured from that particular sample. And after researching that a bit more, this species doesn't appear to be toxic, but it does feed voraciously on phytoplankton, and its personal favorite is Thalassiosira, and this particular species is one that's present within the Mediterranean. So some anecdotal evidence that this might be one of the diatom species that we're seeing in the area, certainly if we're seeing Noctiluca scintillans around. So now we have the annual chlorophyll. We're looking at the latter part of the year, which is this area over here. And I'd like to spend some time analyzing this because we have an inversion again in the photosynthetic pigments. We have a dominance now of chlorophyll. And this, so if we plot chlorophyll against pyrethrin, if I just go through this plot, there's quite a bit of information to take in here, but the red line is the line of best fit for a ratio of 4:1 between pyrethrin and chlorophyll. So pyrethrin 4, chlorophyll 1. And this is the ratio that you expect to find in the marine environment. And then if you look, the blue line of best fit, that is the line of best fit that describes the data that I have collected. And what you see is that there is, the line of best fit describes the data to about an 84% accuracy. Now there are two other lines in that plot. And what's interesting is that the first one is the line 3:1, the ratio 3:1, and the last one is the green one is the line for 1:1. The reason why this is interesting is that the cyanobacteria actually live within that ratio, 3:1 to 1:1. So now we have a chlorophyll-dominated signature, which is in the ratio that we expect to find when we have cyanobacteria within the water column. So that's another interesting observation. And when we look at this graph again, what we realize is that not all of it, but certainly some of it, is in autumn, which is between the 21st of September through to the 21st of December. And when we come back to the plot that we were looking at before, this is, you see that one of the expected algae that we expect to find there are the cyanobacteria, and that has a definite signature in the autumn. So it seems to fit into place with research that we are seeing out there. But a lot of work is required. Unfortunately, these are just preliminary results. It's very difficult to get to compose so much together, especially when we've got so much chemical analysis that needs to be done and so many biological identifications that need to be completed too. And this is ongoing work that we continue to look at in the university, and hopefully we will get some stronger results coming through from that. So in conclusion, we have positive identification of predatory dinoflagellates. We've got Noctiluca scintillans, which is suggestive of Thalassiosira and the presence of that species within the water column. A possible link between the autumnal bloom and cyanobacteria, and Coccolithophores, which have been positively confirmed through microscope work that we were looking at before. So special thanks to the Gibraltar Museum, Dr. Clive Finlinson. I appreciate the opportunity. Dr. Geraldine Finlinson and Stuart Finlinson, thank you very much for having me. The University of Gibraltar, my supervisor, Darren Farr, we've struggled a lot to get to where we are today, so thank you. Dr. Jeremy Young, a specialist supervisor in the UK, who's really helping us untangle a lot of these relationships. And also to my team at the Nautilus Project, Melanie, Brian, Enan, and Alex, who have been instrumental in helping with a lot of this research. So thank you very much for your time and attention.
Very interesting talk. Lewis, do you have questions from the floor? We'll start with Charlie Davis and then Clive Finlinson.
Luis, what I've noticed is that the autumnal bloom in the strait seems to coincide with the massive feeding frenzies when you get a lot of flying fish, a lot of mackerel and the tuna feeding on these shoaling fish. So is there any correlation between the autumnal bloom of plankton and
There is, you're exactly right, Charlie. At the end of the day, it's the plankton that are driving all these feeding frenzies. They're the primary producers, and they're the ones that start the whole food chain off. They kick it all off. So the reason why we've got these feeding frenzies happening now is because there is an autumnal bloom in progress. Okay, although we're probably at the latter end. So it's key to understanding everything that's happening in the rest of the ecosystem. You know, we've been talking about birds, we've been talking about other, and so driving this phytoplankton bloom is driving all of this, and so we really need to untangle a lot of what's happening in the water column to try and contextualize what we're seeing and all the great work that's been done in all these other fields. So yeah, absolutely.
Clive Phillison.
Yeah.
Thank you.
The autumnal bloom in primary productivity does not fit with what happens in the Mediterranean generally, as I understand it. So are you saying that the Strait is behaving more, is it more comparable to Atlantic waters? Is there really a bit of Atlantic coming in?
So, yeah, that's a great question. And I think the problem that I have in my sampling is that I'm only getting down to 80 meters. And so really what we're getting predominantly is the Atlantic water coming in. And so it's that Atlantic signal that we're seeing. However, it's still driving primary production in some way, shape, or form in our area. So sometimes we tend to think, well, you know, we've got the Mediterranean right next to us. We are a Mediterranean basin. But actually, certainly in the surface waters, what we're seeing is that we're probably more Atlantic than we are Mediterranean, certainly just off of Europa Point.
Can I follow up to that? Do you have any indication of interannual variability in what you're describing here?
The primary production yes.
Between years?
Yeah. So
is it consistent or?
It is pretty consistent. What varies is the length of the bloom. So this year we had a very strong bloom. If I were to link it in with other species, we saw the fin whales come through early this particular year. Now, I think that one has something to do with the other. Obviously, more work needs to be done in order to ascertain that for sure. I'd need to repeat this over a number of years to be conclusive on that. But it's certainly suggestive of the fact that there's a link there.
Okay. Any further questions? One at the front, Geraldine Finlayson.
Forgive me if I've misunderstood this, but
Okay.
I just wonder, would it be possible to model what effects the lowering of sea levels would have on these pulses coming through?
Yes, the answer is yes. Okay, it is possible to do it. I'm not a physical oceanographer, so in order to create those models, I would need certainly the help of somebody or the advice of somebody that's better qualified to answer that question than I am. But it's certainly possible to do it using those signatures, yes.
And as a follow-on, do you have any sort of intuition as to what way it would be affected?
Yeah, in truth, I don't. So I tend to read what I see rather than try and interpret with the physics. I have, as you've seen in the presentation, what I have tried to do is fit in the biology into what we're seeing from the physics and from the chemistry. And I know that it's only kind of superficial, but I'm a biologist, so it's easier for me to fit in the biology into that framework than it is to go right to first principles and really break down the physics, which is not really my area, to be fair.
Any further questions? Darren Farr.
Luis, no, I was just wondering, what were your thoughts with regard to what might be driving the changes in dominance between the two different types of photosynthetic pigments?
Okay, so what period throughout the year? The spring, or we're talking about the latter part where we had the flip?
you have a flip about halfway through when it suddenly diverges, and then it falls into line, but one is above the other, which is
Like
so there's a flip about halfway
this
through the year.
part, right? Yeah. So I think that what we have here is I'm not convinced by the May plot. I think the May plot is a little bit that point is a little bit further up, and I think what we're really seeing here is the tail end of the spring bloom. And so what we have are the predators consuming the diatoms. And so what we're getting, if you imagine your your classic predator-prey relationship, the predator always follows, the population of predators always follows on from the prey items. I think we're seeing some signature that's similar to this. In answer to the second part, I think that the flip starts to happen because in July, August, we've now had the thermocline established for a longer period of time. The nutrients are probably limiting. And so now we have the flip taking place because we're starting to see, I would have thought, Coccolithophores in that zone starting to dominate during that summer thermocline period. That's my take on it.
Any further questions? One from Stephen Wall.
I've got a question and an observation to make. The first one is that we're certainly seeing the signature of the Atlantic water in the intertidal habitat, including artificial revetment. Species like Fucus spiralis, for example, which is an Atlantic species, we're finding in our waters. And also the wrecks. You won't find them further into the Mediterranean, but you will find them in Gibraltar all the way up to Estepona. So that's another interesting observation which ties in with the data that you're collecting. My question is, do you think that the bathymetry in the area where you're sampling would have a very significant impact on some of the data you're collecting, specifically thermoclines, temperature, and salinity, given the fact that you are sampling in an area of upwelling?
Well, tying into what you were saying in your talk, it's the thing that struck me exactly about your talk when you were showing that continental shelf area that you were saying was the height of Gibraltar again going down. Because typically where you have such a steep gradient, what you have is nutrient-rich waters coming up. You have some sort of upwelling taking place. Now, I know that the Mediterranean water is leaving primarily, and this is generally nutrient poorer, and the Atlantic water is coming in. But if we have some upwelling there, what we might have is nutrients coming to the surface because of the physical structure which might be helping to drive primary production in that area. It's something that I'd like to investigate more. It only occurred to me today after listening to your talk, so thank you for that. But it's certainly something that I'll be keeping an eye on going forward. Yeah.
Okay, any further questions? No? Thank you very much, Lewis.
Thank you.